Hi everyone, I am going to make a brief video about SNV arrays shown here and the relationship between the logR ratio and the BAF frequency we can analyze from this figure. So to analyze those data, we are mainly using those two formulas, also shown in the slides. Let's start. So first of all, if you start to watch the changes in chromosome 2 and chromosome 14, they are of similar behaviors anyway. You can get the formula which is log2 VA plus VB times C plus 2 minus 2C by 2. This is about minus 0.5. Okay, then in this case, we know that it's a copy number loss, and so from its log ratio of minus 0.5, that we can already know that's a copy number loss. In normal cases, we have A allele is one copy, while B allele is one copy, so they get two copies of allele for each chromosome. But in this case, we already know this is the loss of one copy, so we set VA is 1 while VB is 0. You can try other way around, but it will gives you different B allele frequencies, as we can show later. Anyway, under this setting, we will be able to get log 2C plus 2 minus 2C by 2 is minus 0.5. So 2 minus C equals 2 to the 0.5th power times 2. And this is about 1.24. So we get C in this case is 58%. Which means the clonality of chromosome 2 own cells are 58%. And then we plug into the formula about B allele frequency, we can get BAF equals 0 times C plus 1 minus 58% divided by 1 plus 0 times 58% plus 2 minus 2 times 58%. So if you calculate it, it will be 29.6% for the B allele frequency. And then in this case, because B allele frequency is this one, we can get the allele frequency for another allele. Say allele A is that frequency of A equals 1 minus this value, 70.4%. And then we look at the figure of the BAF, we can see exactly 4 bands, which means B allele frequency 1, 0, about 2/3, and about 1/3, which corresponds to our calculation. Also, we can do similar things for chromosome 14. But the log ratio in this case will be minus 7.5. Okay, so in this case, we solve C. Which is 81%. And then we also calculated B allele frequency. This one is 16%. So we view the band again. We will see that the bands are corresponds to 1, 0, about 80% and about 20%. So this calculation also corresponds to our assumption. So what happens in chromosome 2 and 14? Are they happened in the same cells or different cells? So from those calculations, we can know that 84% plus this value is already bigger than 1. Larger than value 1 means that those behaviors are dependent. So we can sort of draw the behaviors of the two chromosomes. We suppose that clonality of the cell's lost copy in chromosome 14 is about 80%, and then clonality for another is about 60%. For lost one copy in chromosome 2, CHR14. We can get the figure that two cells are normal because the clonality is 80 for both copy number 2 and chromosome 2 and 14. And 80% of chromosome 14 lost one copy. Together with 60% lost one copy. And note that only based on the calculation, we can also draw a figure similar to this. While no cell is normal, but they all kind of getting one copy lost. But since we already think they are higher than 1, meaning they are dependent, so we would rather think a higher probability that they are a subclonality inside a clonality rather than the whole thing getting a variant. Next, we can deduct a similar analysis on chromosome 5. So for chromosome 5, we can see two parts. One part is that the loss of copy at the log ratio is minus 0.75. The other is log ratio is minus 2.5. So based on our previous calculation, we already know that the log ratio of minus 0.5, meaning the clonality about 60, while the log ratio of minus 75, meaning previously those loss of copies happen in different chromosomes, but this time, as we can see on the chromosome 5, they happen on the same chromosome. So, and 60% of which lost in right half of the copy, we denote it as QR. while 80% lost the left copy, we denote as PR. Immediately, we can draw two normal cells, PQ, PQ. And then 80% of them lost one copy of P. But still have both copies of Q. Can do like this. So while others lost the copy of 1Q. So in this case, we may draw lost the copy of 1Q either like this or like this. So we may not know until further evidence came, such as the fishing or pair and sequence. So that's all that I can talk about the slides and the calculation going here. Thank you.